Visual physiology. When light strikes the network of cells that line the inside surface of the eye, it initiates a chain of neural events in the visual system that leads 100 milliseconds or later to a conscious visual experience. The visual system comprises all the neurons responsible for seeing, which can be divided into three major components: the retina, the visual pathway, and the visual cortex. The retina contains the neural circuitry connecting photoreceptors to the ganglion cells whose fibers form the optic nerve. This visual pathway includes the optic nerve, the cell nuclei to which its fibers project, and the onward projection to the cortex. The visual cortex includes all the cortical areas that contain cells responsive to visual stimuli. Of course, this will review the major features of each component: the retina, the visual pathway, and the visual cortex, following the route of neural signals as they travel through the system from the photoreceptors to the cortical cells. Again, if we were to look at the visual system, it includes the retinas, the visual pathway connecting the retinas to the brain, and the visual cortex. We have a right visual field and a left visual field, which go through the temporal retinas. And eventually back toward the visual cortex and the hind part of the brain. Photoreceptor cells in the outer nuclei or nuclear layer, or the nearest to the choroid, are responsible for transducing light energy into neural signals. Ganglion cells in the ganglion cell layer supply the nerve fibers that carry neural signals out of the eye and along the visual pathway towards the brain. And the inner nuclear layer contains bipolar cells, other cells, and horizontal cells. It is complex, and its neural circuitry connects photoreceptors to ganglion cells. Then there is what's called the outer nuclear layer, which is composed of photoreceptors. Then we have photon absorption, photon direction, photon frequency, photoreceptor responses, photoisomerization, graded potentials, and univariance. Univariance, for example, is when a photon is absorbed. Its effect on a photoreceptor is the same regardless of the photon's wavelength. Then the inner nuclear layer, which contains bipolar cells. And of course, think about how later processes could infer wavelength from receptor activity. And we have photoreceptors on bipolars and off bipolars. Key terms would be the cone bipolar, which is a class of retinal cell that conveys activity from one cone photoreceptor to ganglion cells. And then the rod bipolar is a class of retinal cells that conveys activity from rod photoreceptor to amacrine cells. We have other key terms as well. Horizontal cells is a class of retinal cells whose axons spread laterally across the retina, making contact with several photoreceptors. The amacrine cells is a class of retinal cell that has extensive contacts with bipolar cells, ganglion cells, and other amacrine cells. It serves as a number of functions. The ganglion cell is the class of retinal cell that provides the output signal from the retina. Ganglion cell axons form the optic nerve. Midget ganglion cell is the most numerous class of ganglion cell. It responds only at high levels, showing spatial opponency, and may also show red-green spectral opponency. Then parasol ganglion cell is the only class of ganglion cell that remains active at low light levels. It shows spatial opponency, but does not show spectral opponency. Bistratified ganglion cell is the least numerous ganglion cell class. It responds only at high levels of light, has no spatial opponency, but shows blue-yellow spectral opponency. And so we just went over horizontal cells. Macron cells, again, the ganglion cell layer is the layer containing the cell bodies of retina ganglion cells, or RGCs, though it also contains some macron cell bodies. The different types, there are different types of cells, midget, parasol, and small bistratified. Spatial opponency. Spatial opponency is the excitatory responses generated in one part of the retinal receptive field and are opposed by inhibitory responses in another part as well. The spectral opponency is that wavelengths from one portion of the spectrum excite a response, and wavelengths from another portion inhibit a response. And so centers surround ganglion cell responses were first observed in a mammal, a cat, for example, by Stephen Kuffler in the year 1953 at Johns Hopkins Hospital in the United States. And of course, we have a relation between spectral opponency and spatial opponency. Sustained temporal response is a change in response. A change in response is sustained at a relatively high level for the whole duration of stimulation. Transient temporal response is a change in response occurring only at the onset and offset of stimulation. And neural blur, the removal of spatial detail in neural responses as a result of neural processes rather than optical effects. Information processing by ganglion cells, spatial filtering, chromatic filtering, chromatic channel, and achromatic channel. A chromatic channel is a channel of processing in the visual system that conveys information about the chromatic or color properties of the image. An achromatic channel is a channel of processing in the visual system that conveys information about the luminance or light-dark properties of the image. Then we have optic nerve fibers. We have what's called lateral geniculate nucleus, or LGN, which is the mass of neurons in the thalamus, where the optic nerve fibers form the retina terminate. And then, of course, we have the visual pathway, and then, of course, representation of the visual field with and then we have what's called binocular neuron, is a neuron that has a receptive field in each eye, and then partial decussation, which is partial crossover of each eye's optic nerve fibers in the visual pathway, so that half stay on the same side of the brain and the other half cross over. We have what's called magnolayers, structures of the LGN. Again, the LGN is the lateral geniculate nucleus, the mass of neurons in the thalamus where the optic nerve fibers form the retina terminate. LGN layers, we have the magnolayers, which with large cell bodies, they receive projections from parasol ganglion cells, and the parvo layers, which are layers within the LGN with small bodies, they receive projections from midget ganglion cells. And of course, then there's bistratified, but then there's another sublayer of the LGN, conio layers, LGN magno and parvo layers that contain the smallest cell bodies, they receive projections from bistratified ganglion cells. The names attached to different layers are based on Latin or Greek words. Magnus means large, obviously, and parvus means small in Latin. Conus means dust in Greek. Then we have in the visual cortex, we have signal processing before moving on to the visual cortex. So the signal processing in the LGN, or again, as we've discussed, the LGN is the lateral geniculate nucleus. These particular functions have been proposed to sharpen spatial responses by modifying the center surround organization of the receptive fields, perhaps increasing surround inhibition. To begin the process of integrating information across larger retinal areas by promoting synchronous activation of ascending LGN signals, or to enhance the salience of sudden changes in stimulation by modulating the firing patterns of LGN neurons. In terms of the visual cortex, we have the striate cortex, which is so called because of its characteristically striped appearance in anatomical sections. So I guess it would be the striate cortex. The striate cortex is the primary receiving area for visual signals from the LGN. It is also known as V1 or Area 17. Extrastriate cortex, which is areas of secondary visual cortex that receive signals from striate cortex. And then, of course, you have connections to the extrastriate cortex and cell properties. We have simple cells, an orientation selective cell in visual cortex with an elongated retinal receptive field containing excitatory and inhibitory zones. The complex cell is a cell in the visual cortex with a relatively large receptive field that does not contain identifiable excitatory or inhibitory zones, but nonetheless it is orientation selective. And then direction selective cell, a visual cell that responds more strongly to retinal movement in one direction than to movement in the opposite direction. And so those are simple cell, complex cell, and direction selective cell. Concerning binocularity, we'll talk about ocular dominance. Ocular dominance is a property of binocular cortical cells describing their tendency to respond more strongly to stimulation in one eye than to stimulation in the other. Binocular disparity is a small difference between images in the two eyes that provide a cue to depth, usually created by viewing a scene containing objects at different depths. And double opponency is a property of an antagonistic centered surround receptive field in which certain wavelengths evokes excitation in the center and inhibition in the surround, vice versa for other wavelengths. Here it says compare the architecture of striate cortex with the architecture of somatosensory cortex. Here it says key terms is ocular dominance column, which is a slab of cortical tissue running perpendicular to the cortical surface, in which all binocular cells share the same degree of ocular dominance. Orientation column is a slab of cortical tissue, also running perpendicular to the cortical surface, in which, however, all orientation selective cells share the same preferred orientation. And then we have blob, which is a cluster of cells in the superficial layers of striate cortex identified by a staining technique, many of which are color selective. Relative area of the striate cortex devoted to different regions of the visual field. The right half of the visual field is shown on the right, subdivided into zones centered on the fovea. The surface of the contralateral striate cortex is shown on the left, marked and shaded to match the visual field diagram. A very small central portion of the visual field occupies a large proportion of cortical surface. Hypercolumn. A hypercolumn is a block of cortex containing one complete cycle of ocular dominance and a full range of orientation preferences. Then we have what's called response latency, which is the time that it has elapsed between the onset of a visual stimulus and the onset of a response in a given neuron. Response latency. And then we have what's called stripes, which is groups of cells in area V2 of visual cortex identified by a staining technique. Here's two streams theories of cortical function, what versus where, Magno versus Parvo, and perception versus action. We have processing stream, which is a series of neural processing stages in a sensory system, specializing in the extraction of certain stimulus attributes. The ventral stream is a processing stream including cortical areas V4 and IT, which is claimed to specialize in the analysis of spatial form. And then dorsal stream, which is a processing stream that includes cortical areas V3, MT, and MST, which is claimed to specialize in the analysis of movement, depth, and action. And then, of course, we'll just briefly summarize a little bit of what we've gone over. "30 years after the dorsal versus ventral distinction was first proposed, debate continues as to its functions and its significance. Significance in terms of its function. The following observations are consistent with the view that the dorsal stream has a specific function that requires fast processing, but information is shared extensively between the two streams. First, there is a large disparity in size between areas in the two streams. The total cortical area devoted to the ventral stream areas is 2.5 times larger than the area devoted to the dorsal stream areas. This indicates that the dorsal stream has a more specialized and/or simpler function than the ventral stream. There is a marked difference in the latency between the two streams. The three dorsal areas have almost identical fast latencies. Responses in ventral areas are up to twice as slow. This indicates that the two streams do not act in parallel, at least in the sense of response timing. Whatever the dorsal stream does, it has to be done fast. And so, again, the cortical area devoted to the ventral stream areas is V4 and IT. Remember, of course, the area devoted to dorsal stream would be V3, VA, and MT, and MST. There are extensive reciprocal connections between areas forming the two streams, indicating a high degree of crosstalk between them. Of course, fourthly, there is no clear dichotomy between the streams in terms of cell response properties. The full range of stimulus specificities is found in both streams, though in different proportions. Along with the previous observation, this indicates that the two streams are not strictly segregated modules dedicated to processing separate stimulus attributes. They may specialize in different tasks, for example, action guidance, shape analysis, etc., rather than in attribute processing. To sum up the information, the visual system can be divided into three major components: the retina, the visual pathway, and the visual cortex. Concerning the retina, The retina is a layered network of cells lining the inner surface of each eye. The outer nuclear layer contains over 120 million photoreceptor cells that generate graded changes in neurotransmitter release in response to changes in illumination. The inner nuclear layer contains a complex network of bipolar, horizontal, and amacrine cells through which the photoreceptor responses converge into ganglion cells. The ganglion cell layer contains 1 million cell bodies of ganglion cells whose axons form the optic nerve. Midget, parasol, and small bistratified ganglion cells supply over 75% of optic nerve fibers, and their activity carries information about spatial and temporal variations in the luminance and spectral properties of the light striking the retina. Other ganglion cell types are also likely to play important functions, but relatively little is known about them. Okay, of course, the retina, what you need to remember there, about the photoreceptor cells that generate graded changes in neurotransmitter release. All right, in response to light, we're also going to need to remember that the inner nuclear layer contains a complex network of cells. You're going to remember bipolar, horizontal, and amacrine. All right, and of course you're also going to remember the ganglion cell layer. All right, whose axons form the optic nerve. Okay. The axons of the ganglion cells from the ganglion cell layer form the optic nerve. Then you're going to remember also 75% of optic nerve fibers are the midget, parasol, and small bistratified ganglion cells. Concerning the visual pathway carries responses from the retina to the visual cortex. Again, the visual pathway is carrying the responses from the retina to the visual cortex, and it's the median part there. In mammals, for example, the major termination site for optic nerve fibers is the lateral geniculate nucleus, or LGN, in the thalamus. It is located in the thalamus of the brain. The major termination site for optic nerve fibers is the lateral geniculate nucleus, LGN. Due to partial decussation, fibers carrying responses from the left visual field of each eye terminate in the right LGN, and responses from the right field of each eye are Due to partial decussation, fibers carrying responses from the left visual field of each eye terminate in the right LGN, and responses from the right visual field arrive in the left LGN. Each LGN contains 2 magnocellular layers and 4 parvocellular layers. Again, think about magno, large, and parvo, small in Latin, each separated by a coniocellular layer. Parvo, magno, and conio layers receive selective projections from midget, parasol, and bistratified ganglion cells, respectively. LGN cells project to the primary receiving area in visual cortex V1 and to extra striate MT. The preponderance of non-retinal input to the LGN indicates that its main function is to modulate incoming responses on the way to the cortex. I remember we said that conio or conis means dust. All right, and then we have the visual cortex. The visual cortex comprises the major receiving area V1, also known as striate cortex, and the numerous extrastriate areas. Cortical cells respond selectively to stimulus, orientation, movement, direction, color, and binocular disparity. Extrastriate cortical cells have larger receptive fields and more complex responses than striate cells. In all cortical areas, cells are distributed in a highly ordered pattern. The precise function of many extrastriate areas is not clear, but a broad division between 2 parallel streams is a central element in current theories. One stream includes a series of ventral areas, and the other pass through more dorsal areas. Of course, the ventral system, or ventral stream, is thought to specialize in the analysis of spatial pattern and form. The dorsal stream seems to be important for visually guided action and navigation, though its function is still debated. And then, of course, we talked about all different kinds of cells within visual physiology. Next, we're going to talk a little bit about color vision.